So before we start chapter 18, which is the part of the cardiovascular system, blood, I will go over in this unit 4 again. Unit 4 has, if you see here, note, circulatory system, and circulatory system is made up of cardiovascular system and lymphatic system. Okay, and unit 4 we also study the respiratory system. So in cardiovascular system, cardio means heart, and vascular means blood vessel. So cardiovascular system is the system of heart, blood vessels, and the blood inside the blood vessel or heart. Okay. So first thing we're going to talk about blood inside the blood vessel and the heart. Do you think blood is normally found anywhere except inside the heart and inside the blood vessels?
Do you think that the blood is only in the heart and the blood vessels?
Yes.
Yeah, it's a closed chamber. It's a closed circulatory system, right? Is that
Is there any chance that you will find normally blood anywhere in the body?
At
Yes.
an injury site?
That's the abnormal condition. That's a pathology. Normally, no. Normally, you will not find blood outside the blood vessels or heart. Okay. So, in this chapter, we will go over an overview of the circulatory system and blood. Then mainly we will focus on composition of blood. What is blood made up of? It is made up of blood plasma, and inside the plasma you have cellular component, which is called otherwise formed elements. Then we will go over blood cell formation, how blood cell is formed in the bone marrow under the influence of a hormone produced by your kidney called erythropoietin. So bone marrow as the site of hematopoiesis. Hematopoiesis is the processes of blood formation. And then the stem cell of all these blood cells are one. And from that stem cells all divide further in different generation, and they become different kind of blood cell. So we're going to talk about cell line in blood cell formation. And then at the end, we will talk about some disorders of blood like anemia, hemocythemia, polycythemia, leukemia, several other stuff. Okay. So before we start the blood, here is every time you go to doctor, even for a regular checkup, or if you have any suspected infection, your doctor do CBC test, complete blood count. Okay, complete blood count means doctor will, the pathologist will separate the cellular component of your blood. Okay, and find out which one is in what numbers. So here is a clinical case. Your child has had a moderate fever for 2 days. On third day, you take her to pediatrician. After an examination, blood is drawn and CBC, complete blood count, with differential is performed. Complete blood count means what are each component in your blood? What is plasma? What is certain percentage? And differential count is means what are the percentage of white blood cell inside the blood cells or among the blood cells. The question asks, how does this information aid the pediatrician in determining whether the cause of infection is viral or bacterial? So we do complete blood count to find out what is the change in blood component. And based on different kind of change, we can find out whether the infection is cancer, or it is acute infection, bacterial infection, or it is viral infection, parasitic infection, allergic infection, what kind of infestation or parasitic infestation we have. Based on this finding, we can determine. That's why we do complete blood counting. Okay. So blood is a connective tissue. It's a fluid connective tissue. Blood is a fluid connective tissue that is found in the blood vessels. Okay. So here again, cardiovascular system is cardio, heart, and blood vessels. Let's talk a little bit about blood vessels, and I want you to write down this note. When I speak up, write it down. And if you know, that's good. So blood is found only two locations, either inside the blood vessel or in the heart. Okay, in the chambers of the heart. Blood vessel, you can see in the parenthesis in my note. Blood vessels are artery and veins. Okay, so let's see the definition of artery. Artery here. Artery is the blood vessels which carry blood away from the heart, like pulmonary artery, aorta. These are the arteries. Artery always carry oxygenated blood except pulmonary artery. Pulmonary is the lungs. So arteries which carry blood to the lungs are not oxygenated. That's why, again, arteries carry oxygenated blood except pulmonary artery, which carry deoxygenated blood. Now, vein, just opposite of artery. Veins are blood vessels which carry blood towards the heart, which carry blood towards the heart. All veins carry deoxygenated blood except pulmonary vein, which carry blood from the lungs to the heart, and that is oxygenated blood. Again, veins carry deoxygenated blood except pulmonary veins. All veins carry blood towards the heart. Okay. Now, the structure of the blood vessel and artery, artery and veins. Artery is connected to the heart chamber, and it is connected to the pumping chamber of the heart. That's why the pressure in the arteries are higher compared to the veins. Veins are bringing back blood to the atrium, and that's why it has low chambers. The wall is also thicker in the arteries compared to the veins. Okay, so that is the comparison of the artery and vein. I will draw just a simple diagram of any artery and veins in the body. So let's see, this is the artery here, big artery. Then artery divides. You can see they further divide small capillaries, and then ultimately they become capillaries. And then again they join and they forms the vein, small venules, and then large veins. From the venules. Okay. So what is the purpose of any artery in the body? You see, it's a large artery, small arterioles, and then capillaries. These area has the smallest capillaries, And this is the location where blood vessel exchange gas, nutrition, ions, hormones. Okay. And then blood again collected, get into the veins, and then veins drain back to the heart. That's why each and every blood vessels are connected to your heart directly or indirectly. Okay. So,
Professor,
uh-huh.
real quick about the pulmonary vein. You said it carries oxygenated blood to um, the lungs. Is that what you said?
Pulmonary vein, no, pulmonary artery carry to the lungs. Pulmonary vein carry to the left atrium. Okay. So if you see here in this diagram here, artery veins, aorta, then aorta becomes small artery, arterioles, capillaries, venules, and ultimately veins. Okay. And we're going to see about these things a lot in next lecture. Okay. So the blood is powered by pumping action of heart. Function of blood carries respiratory gases like oxygen, carbon dioxide, nitrogen. These all gases are carried by our blood. Nitrogen we don't use, as it enters, it comes back. Nutrients like carbohydrate, amino acid, glucose, proteins, vitamins, and other macromolecules, hormones. This also helps regulate your body temperature. So when you have cold, your blood vessel in your skin constrict and send the blood in the core of your body so you maintain body temperature. And when it is hot, skin blood vessels is dilated and the blood comes in your skin, and by the convection method you lose heat from your body, so you cool down your body and helps body regulate body temperature. uh, a male contains 5 to 6 liter of blood and female 4 to 5 liter in average. uh, the composition of blood, which is the most important part. So as you know, if you take a blood sample of blood and see in a test tube, you see just red, very red liquid, dark red liquid. But if you centrifuge them, you can separate the blood into the liquid portion and the cellular portion. So the blood contains cellular and liquid components. They are a specialized connective tissue. Why we call blood connective tissue? Because it has the cells and extracellular matrix, which contains fluid. So a specialized connective tissue which contains cells, that is blood cells, that's formed elements, and plasma, which is fluid portion, plus fibrinogen, which is clotting factor of blood. In the blood, the most numerous cells are the red blood cells or erythrocytes. And the percentage of red blood cell in any blood volume is called hematocrit. So the definition of hematocrit is measurement of percent of RBC in centrifuge blood, which is 47 plus minus 5 in male and 42 plus minus 5 in female. So higher the hematocrit, that means more the red blood cell you have. Lower the hematocrit, less the red blood cells. And if you have less red blood cell, that is called anemia. If you have high number of red blood cell or hematocrit, then we call them polycythemia. Okay, so here you go. This is the vein. So you put tourniquet, and tourniquet then tightens the veins, return blood to the heart. So when you block it, what happens? Vein bulges. Then you take blood from that vein. If you see in your body, look at your hands, the blood vessel you can see, all the blood vessel, what you see on your surface of your body, they are veins. They are superficial. All the arteries are deep in the connective tissue or in muscles. Why it is made that way? Why we have evolved? Why we are evolved that way? Because that way we are protecting the blood vessel. Because if artery cut, artery is connected to the heart directly. So if artery cut, have you seen anybody cut of artery? The blood comes like jet because you have your artery connected to your heart, particularly with the ventricles, which is contracting and force is higher. That's why whatever blood vessel you usually see outside, they are veins, except some arteries which is found on the surface of bones where we take pulse, like radial artery, temporal artery, femoral artery, popliteal artery. Those are the arteries which are slightly over the bone and they are superficial. Okay. So when you take blood from vein, you see, you withdraw blood and place in a test tube, and then you centrifuge it. Once you centrifuge, due to centrifugal and centripetal force, the heavier blood formed elements settle down on the bottom, and clear straw color fluid, which is plasma, sits on the top. So this plasma is amber color here, straw color, which is 55% of all blood. And then there is red part, deep red part. This is the red blood cells. And in the center, there is a buffy coat, whitey coat, buffy coat, which is very little, like 1% of total volume. This buffy coat and the range of amount of erythrocyte or red blood cell together, we call it formed elements. You can see formed elements. So the formed elements is formed by buffy coat and erythrocyte. Buffy coat contains what? Leukocyte and platelets. Leukocytes are white blood cells and platelets are another fragments of megakaryocytes, which is only less than 1% of whole blood. And then rest red area is erythrocyte, which is about 45% of whole blood. Okay. So now in upcoming slides, we're going to talk about what this plasma contains, what this buffy coat contains, what are the leukocyte, how does erythrocyte look like, what is the function of erythrocytes. That's all we're going to talk about. Okay. So let's talk about the blood plasma. Blood plasma is straw colored, a sticky fluid portion of blood. This is the watery fluid, aqueous fluid, which is approximately 90 to 92% of water. And apart from water, it also contains several dissolved solid particles inside it. And they are ions like sodium, chloride, potassium, magnesium, calcium, these all. Nutrients like sugar, amino acid, lipids, waste, proteins, urea, ketone bodies, name it, all the components you will find inside. And among these nutrients, the proteins are very important because proteins have several roles in our body. So there are three main proteins found in the plasma. They are albumin, globulins, and fibrinogens. Okay. So albumin is the major plasma protein inside blood, which maintain colloid osmotic pressure of your blood. That means this protein hold water inside the blood vessel. If you have lack of this protein, your water will leak from your blood vessel and get into your tissue and can cause edema. Globulins are another proteins, globular proteins. These proteins carry lipid in your body. Okay. Some of the globulin proteins are antibodies, which are the part of immunoglobulins, and they are antibodies. Another protein is fibrinogen. Fibrinogen is the protein molecules which help in clotting your blood. So when there is a cut and you are bleeding, this fibrinogen makes the meshwork of platelets and seal the nick on the blood vessel to stop bleeding.
Will you please repeat what albumin is? It just holds water. That you said?
Yeah, this maintains osmotic pressure. Formed elements are the blood cells, And there are three main types of blood cells. They are erythrocytes. Another name for erythrocyte is
Red blood cells.
red blood cells. Sometimes we call red blood corpuscles. Leukocytes, leuko means white, cytes means cells. This is called white blood cells. And platelets are the fragments of megakaryocytes. Okay. So how we differentiate these blood cells in different types based on their size of the cell, structure of the organelles, and staining property, what kind of color they stain with. Based on that, we can separate them. Okay. So staining of blood cells, acidic dye, when you put acidic dye on blood cell, then they are eosine and they stain pink. Basic dye is methylene blue, and that stains blue and purple. And this dye usually used to paint or stain our white blood cell leukocytes. So here, this is a blood smear. You can see here. See which blood cell looks the most numerous here in this diagram?
Red blood cell.
Red blood cell. So if you see red blood cell characteristic, you see on the outer pore region of the red blood cell, they are darker, and in the center they are lighter, pale, because the red blood cells are like you have a balloon, and the balloon from the both side is pressed. Or you can take another metaphor, like you have a donut without hole in the center. So if you press the balloon in the center, what happens? The balloon is further ballooning up on the circumference, and in the center there is less space, so it looks light pass easily. That's why it is lighter. And outside you have thicker area of the red blood cell. The purpose of this biconcavity of the red blood cell is to increase the surface area of the blood so that it exchanges gases with the tissues. This is the most numerous red blood cell, and we're going to talk in detail about the red blood cell. Another cells, if you see in the red blood cell has pretty much no organelles. Okay. And the size is smaller compared to white blood cells. So these other blood cells has nucleus in their cytoplasm, but red blood cell has no organelles, no nucleus. These white blood cell you see here, it's a monocyte. It has large, the kidney shape or horseshoe shaped nucleus. Here is another lymphocytes, pretty much very least cytoplasm, whole cell is packed with nucleus. Then neutrophils, which has multi-lobed nuclei. And that's why sometimes we call them polymorphonuclear leukocytes, PML. Polymorphs, sometimes we call neutrophil polymorphs. Okay. Then you see small fragments, those are the platelets. Okay. So let's talk about the erythrocyte or red blood cell, or we call in short RBCs. This is the oxygen transporting cells. Its diameter is 7 to 5 micrometer, and the smallest capillary diameter is around 8 to 10 micrometer. So let's see, when aorta from my heart comes to my index finger, now in my index finger it is capillaries. Yes. So diameter of that capillary is around 9 micrometer. And when blood vessel, when the red blood cell pass through that small tiny blood vessel, the size of red blood cell is 7.5 micrometers. So it is like pretty much same size. So if your size is 6 feet and your diameter is like 2 feet and you have to pass through the 2 foot pipe, what happens? You have to squeeze through the pipe. And same way, our red blood cell is squeezed through the small capillaries. What is the benefit of squeezing through the small capillaries? Can you guess it?
I'm sorry, what exactly are you asking?
I'm asking, so when red blood cell pass through the smaller capillaries, the size of the capillaries and the red blood cell capillaries are pretty much same. That's why the red blood cell has to squeeze through the capillaries. What is the significance of squeezing through the
They're all
capillary?
It gets
in one.
That to do with exchanging oxygen?
Yeah, because that way red blood cell has like direct contact. The plasma membrane of red blood cell and the plasma membrane of endothelial cells are in like contact. So there is like only two cell thick layer between the oxygen and carbon dioxide inside the capillaries and in the tissue. And that's why gas exchange is easier. Are you following me? So this red blood cell is most numerous of the formed element. Female has 4.3 to 5.2 million cells per milliliter of blood, and male has 5.2 to 5.8 millions per cubic milliliter. Have no organelles and no nuclei. If there is no organelles and nuclei, what you can deduce from that? That means this cell cannot divide. So once they are born, they're gonna die faster. Like if your epithelial cells are dying, what they do? They take from the basal cells and they divide and makes another cell if they die. But red blood cells once die, they have no organelles, they have no nuclei, so they don't divide. And that's why they can survive only for 120 days. And once they die, they circulate from your red blood vessel, goes to a spleen, and then a spleen recycle them, separate into several other component. Okay. So red blood cell contains no organelles. That's why whole red blood cell is packed with a molecule called hemoglobin, which binds with oxygen. Each red blood cell contains 280 million hemoglobin. Are you following me? And that's why if you have no mitochondria, can you make ATP through the beta oxidation? No, you have to do glycolysis. That's why red blood cell carry all the oxygen, but they do not use it because they don't have mitochondria. See, they just what they do, they are just carrier. They are just carrier. They carry from one point to another and give to the tissue. Okay. So hemoglobin is oxygen carrying protein. The red blood cell has the biconcave shape, and that 30% of more surface area is due to concavity. And why is this important? Because that increased surface area increase the rate of oxygen and other gases exchange. Life for 100 to 120 days originate in the bone marrow. So red blood cell is formed inside the bone marrow. Which bone marrow? Red bone marrow, particularly the bones in the skull, ribs, sternum, hip bones, and the red marrow of the long bones. Those are the location where we make red blood cell from the bone marrow. It exits and then it comes into your blood vessel and circulate. Red blood cell is formed under the influence of a hormone called erythropoietin, which is produced in your kidney. So you can see here, the red blood cell, this is the concavity, both side, you see, concave and concave. That's why thickness is only 2 micrometer, but diameter is 7.5 micrometer. If you take one red blood cell and see inside, you're going to see a red blood cell. And if you see inside, you see the dark area inside packed with dark thing, those all are hemoglobin. Can you see here? You have to see with a real highly magnified microscope. So among those 280 million hemoglobin in each red blood cell, let's take one hemoglobin. Hemoglobin hemoglobin. Hemoglobin has heme and globin. So heme is pigment, color pigment, and globin is protein. So if you see here, what is globin? Protein. Protein are the chain of amino acids. So you see this is the chain of amino acid. And these globin proteins have 4 helix chains. And these 4 chains are named after the alpha and beta. So we have 2 beta chain here, and 2 alpha chain. This is beta 1, beta 2, alpha 1, alpha 2. And in each alpha and beta chain in the center is the heme group. Sometimes heme group here. And in the heme group, so what is heme group? Heme group is another macromolecule. And inside the heme group, there is iron molecule. You see the small red dot? Those are the iron. This iron oxidized or reduced, and that's how they carry oxygen. Okay. So the hemoglobin are made up of protein. That means they are made up of amino acid. And if you have any problem with codon, which is genetic mutation of the codon for these chains of amino acid, then you can suffer from disease. Let's see if you have the alpha chain, this red and purple alpha chain. If there is any parts of the chain lacking or abnormal, then you have disease, and that is called alpha thalassemia. If you have similar problem with a beta chain, then we have beta thalassemia. That means if you have abnormal protein, they will not function the same way. They will have less oxygen carrying capacity. Alpha thalassemia is very common in Asian population. And these are mild type. Beta thalassemia has minor and major. Minor is like mild symptoms, and major has very severe symptoms. In that case, you have a lot of red blood cell destruction, you have severe anemia, and you need the transfusion as a treatment. Okay, so now leukocyte, white blood cells. It is white.
Dr. Shaw, sorry, I have a
Uh-huh.
quick question. Is beta thalassemia found in any particular group of people, like alpha beta thalassemia?
No, it is not so common. It is like European ancestral, there is in them, there is slightly higher, but you can find them in everywhere.
Okay, thank you.
Yeah. Okay. So white blood cells. Let's see the difference between red blood cell and white blood cell. So white blood cells are also formed in bone marrow. And from the bone marrow, they are transported to different location. In early age, your white blood cell, some of them come into circulation, some of them go to your thymus where they mature like T lymphocytes, and other white blood cell stay in the lymph nodes or circulating blood and they mature. Okay. So white blood cells are 4,800 to 11,000 cubic milliliter per cubic milliliter of blood. That's the normal value. And white blood cell protect our body from infection from the microorganism. They are found in the blood vessels and in the connective tissue or in the lymph node reticuloendothelial system. So they can work inside the blood vessel and outside the blood vessel. When these white blood cells exit from the blood vessel and go to the infection site in the connective tissue, that process of exiting from the blood vessel, squeezing from the blood vessel, which is called extravasation of the white blood cells, that is called diaperesis. So diaperesis is circulating leukocyte leave the capillaries and goes to the infection site to fight infection. That is diaperesis. There are two types of leukocytes. When we see under microscope, leukocytes, white blood cells are two types, one with toxic granules in their cytoplasm and one without toxic granules in their cytoplasm. So if there is granules, then we call them granulocytes, sites with granules, cells with granules. Agranulocytes, absence of granules in the cell, that is agranulocytes. Okay, so let's see what are those granulocytes, and then we will use these mnemonics. Never let monkey eat banana. Okay, so here this is the blood, and this is formed elements. Formed elements are made up of platelets, leukocyte, and erythrocytes, red blood cell. Leukocytes, different cell count. So the total count of leukocyte is 4,800 to 11,000 per millimeter of blood. Among them, most numerous are the neutrophils, which is 40 to 70% of total white blood cell. Then lymphocyte is second one, 20 to 45%. Monocyte is third one, 4 to 8%. Then eosinophil is 1 to 4%. And the basophil is the least abundant white blood cells. If you see under microscope, neutrophil, eosinophil, and basophil cytoplasm will have several kind of granules, several color of granules. And that's why we call them all granulocytes. Whereas lymphocyte and monocytes do not contain any toxic granules. That's why we call them agranulocytes. Okay, so let's see here. Neutrophils are the cells which has multi-lobe nuclei. It has granules. You see these small dots. And why we call them neutrophil and eosinophil and basophil? So let's see here. I'm going to explain, and I'm going to come here. So eosinophil is, eosine is acetic dye. You know, we talked about earlier slide. Eosinophil is an acidic dye which stands white blood cell. So eosinophil is attracted to acidic dye towards eosine. That's why we call them eosinophils, attracted to eosine. white blood cell attracted to eosin. Basophil is attracted to basic dye, and that's why you call them basophils. Whereas other cells are attracted to both eosinophil and basophil. That's why we call them neutrophil, because they are attracted to both an acidic and basic dye. That's why you call them neutrophil. Okay, so name neutrophil, eosinophil, and basophil is based on what kind of stain, whether acidic, basic, or both, they stained with. Okay, so here neutrophils are stained with both kind of dye. So you see purple and pink both here, and then you see the stained toxic granules inside, and their nuclei are multi-lobed. So this is most numerous white blood cell. They phagocytose and destroy bacteria. So if you have any bacterial infection like pneumonia, the streptococci, throat infection, pharyngitis, sinusitis, any bacterial infection like pneumococci, rods infection, those all bacterial infection will increase the number of white blood cells. So when we do CVC complete blood count, and if there is absolutely neutrophils 90%, then that is for sure some bacterial infection. We don't need to worry about virus. We need to start treatment with the antibiotics. Okay. So nucleus has 2 to 6 lobes, and the granules pick both acidic and basic stains. You see. Eosinophil is only 1 to 4% of all white blood cell, and they play a role in indic allergic reaction or parasitic infection. So neutrophil, bacterial infection, whereas eosinophil, you see here, it is like dumbbell shape. What is this called? Bilobed nuclei, and that is helping in allergic reaction or parasitic infection. Let's see if you have worm infestation, you're going to see this increase in number. Basophils are about 0.5% of all leukocytes, and they usually 2 lobes, their nucleus, and their granules, you see the small dots, granules secretes histamine, and histamine released in the tissue which increase the diameter of blood vessel, and that's how histamine increase the red patch or erythema in your skin. That's why histamine cause sometimes allergic reaction, and we need antihistamines. They function in inflammation mediation, and their function is similar to mast cell, because mast cell also release the histamines. Now, agranulocytes are lymphocytes. They are composed 20 to 40% of white blood cells, and you see their nucleus is large, stained dark purple, and they are effective in fighting the infectious organism, particularly the chronic infection and virus infection. But these white blood cells, lymphocyte, act against specific foreign molecules. When you take COVID vaccine, this is white blood cell, particularly lymphocyte, which takes that antigen and make antibodies and keeps the memory of that particular antigen. If you get infected with COVID next time, these are the white blood cells which protects you. Okay. 2 main classes of lymphocyte, T cells and B cells. T cells attack foreign cell directly. This activated T cell come in contact with the bacteria and kill it. Whereas B cells produce antibodies, then antibodies attach to the bacteria or infected cells, and then T cell comes and other phagocytic cells comes and attack them. Okay. Monocytes is another agranulocyte, which is composed of 4 to 8% of white blood cells. They are the largest among all the leukocytes. You see the big one, and they have kidney-shaped nucleus, and these are the major macrophage, big eater. These are the wandering cells in the tissue too. Okay, they are phagocytic cells. So they are found in your blood vessel, or they can move from blood vessel and live permanently in your tissue. For example, you have a brain tissue and you have macrophage in the brain. What is the brain cell macrophage called?
No, sorry, it's microglia.
Microglia. In the skin, you have Langerhans cells. In the lungs, you have alveolar macrophage. In the liver, you have stellate cells, or we sometimes call them Kupffer cells. They all are these monocyte-like cells. Platelets are cell fragment break-off of megakaryocytes, and the main function of platelet is to clot your blood. So when you have a cut, these are the platelets which get activated. They attach to it other, seal the cut, and then fibrin comes and make it stronger and seal and heal the bleeding. Okay. So here is summary of formed elements. All. What is the cell types? What are their illustration, description, number of cell per milliliter of blood, duration of development, lifespan, and functions. Okay. Just before the exam, you can go over this one 20 times in 1 hour, and you can remember everything. Okay. Now, blood cell formation. Blood cell formation is hematopoiesis, which is process by which blood cells are formed. There are 100 billion new blood cells formed each day. And this is erythropoietin. We talked about this. Bone marrow, they are produced in red marrow. And in the bone marrow, there is yellow marrow. That does not produce any blood cell. They contain fat. Okay. Red cell, after time, they are dead, and dead red blood cells are recycled and processed by your spleen. Cells line in blood cell formation. So here you go. Let me talk about. You see, there is in the bone marrow, you have blood stem cells, and then there is 2 generation immediately after that. So B and T lymphocytes are separate gene from the very beginning. You see, and then there is myeloid stem cells. Myeloid then differentiate into proerythroblast, myeloelastoblaст, monoblast, and megakaryoblast. You will not be tested in these intermediate names, but you remember how they are formed. So proerythroblast forms erythrocyte. You see, at the beginning, red blood cell has nucleus, but slowly they take it out and becomes anucleated. That's why if you see newborn baby blood and check their red blood cell under microscope, you will see nucleated red blood cell because they have some fetal red blood cell. Fetal red blood cell will have this nucleus. Then you have eosinophil, basophil, neutrophil, and then monoblast. So monocytes, and when they are outside the blood vessel in the tissue, then we call them wandering macrophage. Megakaryoblast, you see, big cells. That's why you call them megakaryoblast, and then becomes fragments. Lymphoid stem cells, B cell and T cell. When you see cancer name, blood cancer name, you're going to see a lot like myeloid, lymphoid. What kind of they are? Like lymphoid, lymphoma, leukemia, or myeloid leukemia. Based on that, they are named all from which origin they are coming from, all the cancer of blood. Some other disorders are polycythemia, abnormal excess of erythrocytes. If you have a lot of red blood cell, that is polycythemia, and it is dangerous because increase red blood cell, increase viscosity of blood, that can plug your small capillaries and reduce the blood supply to the tissue, or viscosity also increase blood pressure. Anemia is erythro level or hemoglobin concentration low, that will reduce oxygen carrying capacity. And disorder of erythrocyte are others like sickle cell disease, which is inherited, and that also distort your erythrocyte and reduce the carrying capacity of your red blood cell. Hemochromatosis is the inherited disease, and that absorb a lot of iron from your GI tract, and that can help build iron buildup in your organs like liver, pancreas, and other organs. Disorders of leukocyte like leukemia are a form of blood cancer, and they can be, as I said earlier, lymphoblastic or myeloblastic. Let's see here. So if they are differentiating and become abnormal from this kind of cell, then we call them lymphoblastic. If they are coming from this one, then we'll call them myeloblastic lymphoma, myeloblastic leukemia. Platelets is thrombocytopenia, where thrombocytes or platelets are low. And if you have low platelets, what will be the problem? You will have easy bleeding, like you are brushing and you will see bleeding in your mouth all the time. That is thrombocytopenia. You have purpura or skin spots, red spots in your skin due to the lack of platelets, which prevents bleeding. Okay, so these are all in the blood. If you have any question, you can ask me.
Professor, I wanted to ask, you said something during the class. You said we don't have to worry about the virus.